name's Malcolm Horne, and this is, as many of you will know, is part of a rolling series of lectures about Parkinson's disease, which is directed and intended for people who are affected directly or indirectly by Parkinson's disease, and to keep you abreast of what's happening in the research world and also in terms of clinical management. So tonight's topic is what causes Parkinson's disease, and I think the first point is to state we don't know. So we could finish very quickly here and move on, but I think that there are a lot of clues that we have, and so I'm going to take the position of trying to put it out to you from a historical perspective and help you understand what we do know from these clues that have given to us over what is nearly 200 years. So these are the approach I'm going to take. So I think to begin with, we actually have to have a think about what is a disease. It sounds quite obvious, and many of us think of it as about the idea of infectious diseases like smallpox. So the idea with smallpox is if you catch the virus, you get the disease. If you have a vaccination, you can stop the disease. And so it's very easy to think of this in terms of there being a single causative agent that you can get rid of. And so very often in conditions like Parkinson's disease or Alzheimer's, or we have the idea of we're searching for the cure, with the idea that there will be a virus or a something or other that we can find and fix it, and that will be the end of it. However, degenerative diseases are different, and I'm going to use the example from cardiac disease, which is another degenerative disease, to actually help us understand. So in the 1800s, there wasn't such a thing as cardiac disease. There was a condition called dropsy, and this was where people accumulated large amounts of fluid all over their body, and the cause of this was not known. And in fact, the term was used, which is one we don't use any longer. Some way along the way, people discovered that in fact, the cause of dropsy was in fact a problem with the heart or a problem with the kidneys. Over time, we began to then understand that what caused heart disease was damage to the heart muscle through a heart attack or trouble with the valves. And eventually that led to an understanding that these things were often caused by atheroma or arteriosclerosis or hardening of the arteries, depending on how you want to cause it. And eventually we came to the conclusion that actually all of these things were caused by risk factors. And so there's a sense in which we can think of this progression as going from risk factors to what's really the disease onset, and then the active disease when people have heart attacks or overt symptoms, and then finally the problem of the failed organ. Now, the reason I'm putting that together is that as distinct from smallpox, that means that there is not a single cause. It's actually, really the beginning is this set of risk factors, none of which is probably enough to cause heart disease. And certainly if you have them, it doesn't mean you will get heart disease. But as they come together with a confluence of them, perhaps if you have hypertension, diabetes, smoking, your chances go up and up until eventually you have this process of the changes in the artery. And if you have then, eventually you'll get this problem. And so when we have this process of discovery in neurodegeneration or any form of degeneration, we always begin up here with this idea of being able to find the failed organ, and that's the end, and we have to travel backwards along this process in the same way as we did with heart disease to find this, then this, then this, and then this. So in Parkinson's and many neurodegenerations, we're still up at this end, trying to understand this, and we're working backwards. So let's trace the history of how we got there. So it's almost exactly 200 years ago when James Parkinson wrote his now famous monograph. Next year it'll be exactly 200 years. And what he said was, he called it paralysis agitans, and it was only sometime later that it actually got another famous neurologist A French neurologist actually referred to it as Parkinson's disease. And he described this entity, which I'll show you this video, which many of you would recognize, but he described involuntary tremulous motion with lessened muscular power in parts but not in action, meaning that people could actually move, but they didn't move properly. And even when supported and with a propensity to bend the trunk forward and to pass from a walking to a running pace. And we would still probably recognize that as being Parkinson's disease, and we mightn't use the same type of language, but that's pretty much what we teach medical students. But the one thing he probably got wrong is this last bit, is because we now understand that in fact that's not entirely true, but there is some injury to cognitive function over time. So where are we up to if we use our analogy of or example from cardiac disease of moving backwards? So what Mr. Parkinson's described was the failed organ. That's the problem when you reach that approach of injury, because by the time you get Parkinson's disease, something like 70% of the nerve cells in the substantia nigra are already injured. So it's well down the process of going this way by the time problems actually occur. And if we were to understand what was happening in active disease, we would need to be up there when we were understanding what happened to 20 or 30% of nerve cells being injured. And at that stage, we really don't know what that looks like. We might be able to recognize it if we had more information, but at present, we're really only recognizing these symptoms. So things moved slowly in the world of research. So it went from 1817 to 1895 before the next discovery, when a Frenchman called Emile Brissot discovered that if you looked at this part of the brain, basically if you put your finger in your ear and in your eye and they met in the middle, that's where you'd be. And he found that in fact in people who didn't have Parkinson's disease, there was this area of black material, which is called the substantia nigra, which is what doctors always do, find a fancy name to say the obvious, the black substance. And then he noticed that in Parkinson's disease, that region is no longer black. He also noticed that when 50 to 70% of this area a lot is when symptoms start. So that put us somewhere into the idea of thinking that losing cells in the substantia nigra must be active disease, but it's very hard for us to find that early stage of the process. The next big step was in 1912 when a man called, a neurologist called Louis, found that in those same cells that Mr. Brissot found, the substantia nigra, this is one of these cells, and he found these pale, round accumulations with an edge around them and a translucent appearance in the middle. He didn't know what they were, but since then they've always been called Lewy bodies, and they are the defining pathological feature of Parkinson's disease. If you can't find them in the substantia nigra, it's not Parkinson's. And that's sort of been pretty much the truth we found. So understanding what these were seemed an important clue. The next big step was when these two gentlemen discovered that dopamine was in the substantia nigra, and that if you gave a precursor to dopamine to people with Parkinson's, there was an improvement, and they got the Nobel Prize for discovering that. So that led us to understand that Parkinson's involves damage to the substantia nigra, something to do with accumulation of these things called Lewy bodies, and that the loss of dopamine, which is made by these cells, is important in the production of the symptoms of Parkinson's disease. So you can see that these are clues that are going backwards, is that dopamine's important, Lewy bodies somehow rather feature in the picture, and losing substantia nigra cells is what produces many of the key symptoms we first recognize. The next big step, which has really been one of the great breakthroughs in Parkinson's disease, was two discoveries that followed within a year of each other. The first is by this group of people who found that in a very rare example of a familial form of Parkinson's, in fact they described it in a Greek family, but it is very, very uncommon, but they found that it was due to a mutation in a gene called of the gene that makes a protein called alpha-synuclein. And no one really knew what alpha-synuclein was before then. The only time the description was that it had been found in the brains of birds, and that was really not known. But then a year later, these gentlemen found that alpha-synuclein was the key protein in Lewy bodies. And so suddenly everybody became interested, because everybody who had Parkinson's disease was having an accumulation of alpha-synuclein in the Lewy body. And so suddenly people became seriously interested in alpha-synuclein and its behaviour and pathology to understand what was going on. And so I'm going to talk a little bit about that now in detail. But what that means is, and I've taken you through all of these clues that we've got, and it leads to some questions. So what does alpha-synuclein do? Does its misbehaviour cause Parkinson's, or is it a bystander? And if it is, what causes it to misbehave and get in trouble and participate in Parkinson's disease? What actually is the Lewy body? And why are dopamine cells susceptible? So these are questions that researchers suddenly got interested in after in the late 90s when these discoveries were made, and in many ways they're still the things that are preoccupying scientists now to try and understand what's going on in Parkinson's. So to understand this, and some of you who've been to these lectures before will have seen this bit that I'm going to go through, but to understand this I have to give you a very brief introduction to the biochemistry of the cell. So bear with me as we take this little detour. And this is a cartoon of the cell, and these are the functions which we can describe a bit like a little city around it. So within the centre of the cell is called the nucleus, and the nucleus contains the genes, and genes are responsible for producing protein. So genes are the blueprint about how you will make proteins, and I'm going to talk a little bit about that later on, but they are, if you like, and I've put them there as being the protein template. You have to have those to actually read these blueprints in the factory, which is the thing just outside where the gene is read, and the information is sent to the protein factory, which we call the endoplasmic reticulum, and that makes the proteins ready for being sent out or exported to the rest of the cell and out into the rest of the body and to be used in the appropriate way. To do all of this activity, you need a powerhouse, and they're called the mitochondria. And like all sources of energy, if you can harness it and direct it in the right direction, it's a very useful thing. But the byproducts, if they get in the wrong direction, are quite damaging to the cell. And mitochondria are always producing molecules called free radicals. So they work by taking oxygen and turning oxygen into energy. And the byproduct of doing that is this thing we call free radicals, and they can damage proteins and genes, and so they have to be harnessed properly. And misbehavior of these can end up causing injury to the cell. Now finally, you need a garbage disposal system, because if you're constantly making proteins, then they need to be recycled and reused because they wear out, and the cell needs to be efficient and reuse these pieces. And the garbage disposal system actually turns out to be a key part of what we think goes wrong in Parkinson's disease. And in fact, we think all of these processes, particularly these ones here, are players in some way in the process of Parkinson's, and I'm going to explain that in a bit more detail. We also know that there are some genes that are involved in Parkinson's, and in about 10 to 15% of people, it's actually disturbances in the blueprint that is the origin as well. So let's go through those in a bit more information. So what we think happens with alpha-synuclein, it's normally a protein. We don't still actually know what it does, but we think that it's got something to do with handling the little packages which are wrapped up for export out of the cell. So when proteins are made, they're packaged up in a little bag of membrane called a vesicle, and that's the way in which cargo is sent out of the cell. And one of the important ways that that happens is the way nerve cells speak to each other by releasing a little package of chemicals such as dopamine or other transmitters, as we call them, so that cells can talk to each other. And alpha-synuclein seems to be very important about how these little bags, vesicles, dock to be sent off to foreign parts. Still poorly understood, but it seems that most of the time it sits within the cell as a relatively unfolded protein and that's able to be highly soluble. Under normal circumstances, when this is damaged, it can be removed through a disposal system, a garbage disposal system, and I talked about that as being important in Parkinson's. And this is designed for clearing it when it's in a soluble or single form. But if something happens, then somehow or other alpha-synuclein becomes insoluble. And we're pretty certain that in its insoluble form, it's got something to do with Parkinson's occurring. And we don't know whether it's the bystander and therefore the canary in the mine, as it were, pointing to why there's trouble, or whether it's actually the key culprit. But somehow or other, it seems to be very close to the action of what's causing the problem. We also know that the body very quickly tries to sequester this insoluble form into an aggregate called the Lewy body. So we think the Lewy body is the toxic dump, which has got a membrane around it to keep it out of harm's way and make it less toxic to the cell. And so providing the toxic dump is working and not getting too big, then all is well. But when the toxic dump overflows, we think that that's what causes this thing to become insoluble, to its insoluble component to become dangerous. So I want to talk a little bit more about the garbage disposal system, because this is helping us to understand how there might be ways of managing Parkinson's disease. So I mentioned before that the garbage, if you like, that has to be disposed is damaged proteins and other molecules, of which alpha-synuclein is a very important one, and damaged cell parts, but particularly those mitochondria, the energy factories. And when they become damaged, then they need to be recycled and turned over again. And any poorly made protein, anything which the factory does badly, and that might be because the blueprint is damaged, in other words, there's a genetic fault. But for whatever reason, if there's poorly made protein, or there's damaged cell parts, or there's damaged protein, then they have to get formed into little mini Lewy bodies, little aggresomes we call them, little lumps of aggregated proteinaceous material, and they go into a bag, a vesicle, called the lysosome, which is filled with acid, and that then the protein gets broken down and then is recycled for producing new protein. So this is in fact the sort of the recycling plant of the cell, where it damages and removes protein. Now you can imagine that if there's too much garbage, this system is going to be overloaded, or if this process becomes inefficient because it's damaged in some way, then you're going to get overloaded. We suspect that both things are actually happening at the same time. And we are unclear just how that occurs, but we do know that with age, as we get older, we have nerve cells for your whole life. And as you'll be aware, housekeeping is very important. And if you have a garage out the back, which you don't keep throwing things out, then the longer you live in the house, the more the garbage accumulates and the more that garage is no use. We think the same sorts of things happen with age, that the capacity for renovating and removing becomes less efficient. So the longer you live, those nerve cells you've had all your life tend to be less efficient at housekeeping, and so you get slowly aggregation of protein. There's also some suspicion, but no smoking gun to prove it true, that environmental toxins are responsible for aggregating and damaging proteins more and also interfering with the garbage disposal system. We've known since about 1970 that Parkinson's seems to be more likely in rural regions which use insecticides, and so there has been this suspicion for many years that they may be culprits, but we've never actually been able to prove that that's the case. And so, for example, in Australia, the northwest of Victoria, the Goulburn Valley and the northwest of Tasmania are hotspots for Parkinson's. They're all places which have used large amounts of insecticide, and you'd think, well, there you go, there's the explanation. But on the other hand, the age of the people living in these areas is 15 years greater than in the cities, and it may simply be that what we're seeing is that older people live there and it's not actually the insecticides at all. But nevertheless, there is this sneaking suspicion and worry that there is something about this relationship between toxins that may not be the cause, but an aggravating cause with other factors being present as well. But I want to stress that that has not been proven. It's just a suspicion. We also think that dopamine itself might be something that causes the susceptibility. So it doesn't actually cause Parkinson's, but it might be why the substantia nigra is the first set of cells to become affected in Parkinson's, and therefore is the marker as to why we get the symptoms of loss of dopamine early on. And that's because it's a highly oxidative molecule. And remember, we talked about oxidative byproducts as being a cause of damage. And so its circulation in the cell might hasten the process of these aggregation and misfolding of proteins in some way. And I've also talked before about genes, which I'm going to come to in a little bit more details in a minute. But one of the other questions is that if we have an increased demand for raw materials, then we'd need to actually make this process more efficient. Whereas we have a suspicion that if that demand for raw materials isn't very high, this whole process gets turned down and becomes lazy, and so a risk of accumulating. So one of the things that we know is that if people starve, then they turn on this recycling material and they start using up the furniture as well. So they start dismantling some of the structures within the cell to provide more protein to recycle. We also know that if you take experimental animals and calorie restrict them, then it's much harder to give them models of neurodegeneration. There's been a number of people who have tried and argued that if we were to calorie restrict humans, that we would also have the same effect. And so there is this, again, like the question of insecticide, a long-standing suspicion that the other side of lack of calorie restriction, that is obesity and lack of exercise, may be a risk factor involved in Parkinson's. So that's led to this question around the diabetes model. And the evidence that people who have diabetes are more at risk of Parkinson's is really quite a vexed question. There's some studies which show it does and some studies which show it doesn't. But a more interesting thing has come about is that exercise, which we know turns on the same genes that are helping to prevent diabetes, seems to be important in Parkinson's. And we know most about this in animal studies, where again exercise will prevent models of Parkinson's from occurring. We've not ever been able to show that it's really true in Parkinson's disease with humans. We know though that exercise turns on the same molecules and that are important and protective in animals. But probably what's really most interesting is that there's been emerging evidence that diabetes drugs might help. So there was a chemical called exenatide, which is part of the GLP-1 inhibitor family, and it's used in diabetes. It's a relatively new drug. And last year, a team in the United Kingdom did a preliminary trial of about 50 people in which one half was given exenatide and the other half was given injections, because this is given by injections, but without the active ingredient, and neither the patient or the doctor knew who was getting what. And they did this for 18 months and then looked at people at the end of this study, and they found that the people who had had exenatide had not progressed as fast as those who didn't have exenatide, and the effect was both on movement and also on cognitive function. So this has now led to a whole string of studies which are actively occurring around the world, in which these drugs in this family are being actively trialled to see whether they might have an effect on preventing the progression of Parkinson's. So apart from this being an interesting possibility about one way of managing it, it's also led us to this thinking that perhaps there's more evidence that taking steps to do what exenatide might do, which is to encourage weight loss, turn on the mitochondria, turn on the autophagy, the garbage disposal system, and make it work harder, might stop things like the Lewy body from accumulating and being a problem. And so our general advice to people, although we can't say hand on heart this is work, that if you have Parkinson's disease, it's worth exercising, losing weight, and directing yourself to having a BMI that is the right range. It's probably not a bad idea if you haven't got Parkinson's to think about doing the same thing for all sorts of reasons. So we talked before about the other possibility is that genes might be important in Parkinson's, and we certainly know that a mistake in one of the genes could probably explain up to 10% of Parkinson's disease, particularly in those people who have young onset form of it. But it may actually explain a much larger amount, and I'm going to talk a little bit about genes first and then come back to the examples of what we know in Parkinson's. So gene language is a 4-letter alphabet. There's not actually alphabet land as alphabet soup floating around the cell like that, but they're the words we use for the chemicals in the gene, and to actually explain what they are. And so these three of these letters code for an amino acid, and protein is a string of amino acids. So for example, those 3 letters of the gene alphabet would code for the amino acid called leucine. And so you can string these alphabet words together, but if there was a mistake in the alphabet or in the spelling, we might get various forms of errors, and you can think of this of using our own alphabet. So if there was a word like "let there be peace" and there was a minor spelling, you'd get the wrong meaning and function, but it mightn't be absolutely damaging for your comprehension of what was there. You would just notice that it's spelled wrong, but you would get the idea of what they're really trying to say. And it would just mean there was a bit of inefficiency or clumsiness about how that, and if that was a protein, that would mean that you'd still get the function, but it mightn't be as efficient or as effective it would be otherwise, and so a slightly less useful bit of that protein. But if you had something like this where there was a complete mistake or the words were jumbled, you would lose the meaning and you wouldn't know what was there, and there would be a loss of meaning. In other words, if it was protein language, there would be a loss of function of that protein and it just wouldn't work. So when we speak of mutations, what we're talking about is a spelling mistake. So that means that a mutation can have the effect of impairing the function but not removing it completely, just making that protein less effective, or it might be more serious and stop that protein from working entirely. So in Parkinson's, what do we have? Well, the original alpha-synuclein family that's from Greece is an example where there was a serious mistake in the alpha-synuclein and it really didn't work very well. And they're rare. We have a few examples. They're famous because it gave us this insight to understand what might be happening in non-genetic Parkinson's. But actually, as a cause of Parkinson's, serious mistakes in the spelling are uncommon. What we think is much more common is examples where there's a mild impairment in the spelling. And I mentioned before that about 10% of Parkinson's might be in this, and they're usually of people who have early onset Parkinson's. So there are some forms where you see it in teenagers. There are other forms in people in their early 20s who get it, and we're usually suspicious that it's one of these. However, we think that these might explain maybe even as much as 50% of Parkinson's. We're not sure about that, but we do know that in particular racial groups that there are examples of this. So example, in people of Ashkenazi Jewish heritage, there are two genes. There's one called LARC2 and another one called GBM. It doesn't really matter, just trust me that that's important for their name. But having this type of spelling mistake in those puts you at risk. Doesn't mean you'll get it. So you can have two people in the same family, one has this spelling mistake and the other one doesn't. Both of them have this spelling mistake, one gets Parkinson's and the other one doesn't. But on the other hand, if you look at that family, they have far more people in it with Parkinson's than other people do. So we can think of this as an example, as an analogy. They're not saying it's the same thing, but an analogy with diabetes. If you look at families with diabetes, you can think of families where there's only one person in the family with diabetes. We suspect then it's not any genetics, it's probably all environment. You can look at other families where it's really common for them to have, perhaps a third of the members will have diabetes, and we suspect then that genetics is playing a part in some way. And the last type we want is, again, using a diabetes example, is where we have a gene environment. So in the case of people in the Cook Islands, before Europeans arrived and bought the Golden Arches, it was really rare to have diabetes because they had developed a genetic makeup which made them very well adapted to having a protein-rich and calorie-poor diet. In other words, they had lots of fish, but didn't grow much that it was calorie-rich. When the Western diet arrived with all its calories, suddenly they had a very high chance of getting diabetes, and something like 70% of Cook Islanders have diabetes. So that's an example where the gene doesn't cause diabetes, but the gene in this environment causes diabetes. Now, we don't know whether that occurs in Parkinson's, but we have a suspicion that this might be why insecticides is important. That if you have the wrong genetic makeup, or a genetic makeup which suits you very well to be living in one environment, but doesn't suit you very well to be presented with an insecticide, might put you at risk of Parkinson's. Now, we don't know that, but that's a suspicion. So, in terms of where we know where the genes lie in Parkinson's, when we look at people with Parkinson's, look at their genetic makeup, we see that genes that are involved in agrosome function, making these things, lysosome function, and mitochondria disposal feature much more commonly. Again, leading us to suspect that these things that are involved in some way at making this process less efficient, partly because of the way, if you like, those mild spelling mistakes tip you in that direction of these things going wrong. Why is that important? Well, if they're bad spelling mistakes, it's really hard to fix them. But if they're mild spelling mistakes, it means that they are what we call in the trade druggable. That means that you can, a chance of being able to design a drug that overcomes that problem. And so that's the reason that scientists are very much interested in what's going on there, because it creates what we call drug targets, which then means that you can focus that on there and turn that process around. And in the case of that drug exenatide, which I told you about, part of its action is to make this process more efficient. So one of the other questions that comes up is, so how do we get the Lewy body? How does it start? Why do you have to wait until you're 50, 60 or 70 for it to begin? So one of the interesting things is that if we look at how Parkinson's starts and where it begins, one of the very early things that we know about Parkinson's, that when you diagnose it and you ask people about things like do you have trouble with constipation or controlling the blood pressure or your sense of smell? Significantly more people with Parkinson's have constipation and impaired olfaction than people of the same age, leading us to, and we know why that is, and that is because there are Lewy bodies in the nerve cells in the gut and in the lower part of the spinal cord that control the structures that regulate the bowel. We also know that many people at the time of diagnosis have a type of sleep disorder which is called REM, rapid eye movement sleep disorder, where they act out their sleep, out their dreams, and that wakes, they toss and turn, wakes up their bed partner, who often gets a kick in the shins or because of these vivid dreams that are occurring. And that has already been going on 5 to 10 years before the Parkinson's is actually declared. And then the one that actually draws attention to going to the doctor is when the substantia nigra is affected. And then as the years go by, these other things often, in many cases, become more prominent. And that leads us to an idea that perhaps there's a spread of Parkinson's, of the problem, through the brain like that. Now, a pathologist called Brach from Vienna in 2002 reported some studies that he had done, in which he had done post-mortems on many, many people who had died and then gone to the morgue, and he had looked at their nervous system in a great deal of detail. And what he found was that everybody who had Lewy bodies always had them in the gut and in the part of the nervous system that leads to axons to the gut. Or they would have it in the gut plus this part of the brain that would actually normally be affected in controlling REM sleep. Or they had it here, plus the substantia nigra, and they were the people who had Parkinson's. And so what he was able to put up is this hypothesis, is that it seems like the alpha-synuclein problem begins down here first, and then you see it here, and then it spreads. Or you see it actually in the olfactory system at the same time as the gut. And so this has led to the hypothesis that somehow or other that Parkinson's begins by some disturbance which might be in the environment and gets in and disturbs these cells first, and then there's some mechanism that leads to spread throughout the brain. Again, it's a very interesting and important hypothesis. We can't discount it, but it's very hard to see, and a lot of interest is going on, particularly in this institution. There's a colleague we work with from Andy Hill, who's now moved to La Trobe, and he's been able to demonstrate some of the mechanisms that could be explain how alpha-synuclein spreads from cell to cell and migrates through this process. So where are we up to? Well, we said James Parkinson's told us that. Over the period of time, we've now got all of this information that might tell us how active disease begins, or what it occurs and what's required. You need all of these things: Lewy bodies, substantia nigra cell loss, dopamine in the substantia nigra, L-dopa to people, mutations, and alpha-synuclein is the main protein. We know about Brach's hypothesis. But if we're wanting to actually get further upstream, what we're going to need to know is biomarkers that tell us that the person has Parkinson's or is getting that process. Now, a biomarker, what do we actually mean by that? So a biomarker is a marker that shows that the disease is present. So in diabetes, a biomarker is a high blood sugar. So if you didn't know you had diabetes and you went to the doctor and they took some blood and they said your blood sugar's high, that would be a biomarker that says, despite you not knowing it, you actually have diabetes. So if we had a biomarker, I can't remember what slide's coming next, yes. If we had a biomarker, say, where people who didn't have Parkinson's, they had high levels of it, and people who had Parkinson's had a low level, or perhaps another one, biomarker B, where it was the other way round. And at this stage, when people actually came to the doctor and Parkinson's was diagnosed and that biomarker was already positive, then we could guess that it must have been positive at some stage earlier on in the disease. If we think of this as being normal and the years going by, and these are the nerve cells slowly disappearing and getting less and less, by the time there's only 30% less, you turn up to the doctor and the biomarker is there, then we would be able to guess that sometime along here that biomarker became positive. So if we could go and look at people over a large number, then we might be able to identify those that the biomarker positive and then see whether that helped us understand what do people with early disease look like. That's by trying to find this group here. So we have two biomarkers at present that we're interested, both of them were developed here at the Florey. One's a blood biomarker and the other is a device for measuring movements on the wrist and telling us that, diagnosing the presence of abnormal movements. And we know that by the time you first turn up, this one, patients have low movements compared with people who haven't got Parkinson's, telling us that this must have been going on for quite some time beforehand. We also know that blood biomarker is high compared with those who don't have Parkinson's, telling us that it must have gone high sometimes earlier. So here's the problem. In people over 60, approximately 3 in 100 have pre-symptomatic disease. Now, we don't know how long that pre-symptomatic stage is. We have a suspicion that it's somewhere between 6 and 10 years, but we don't know. So if you think about that, that leads you to say, well, how many people would you need to look at? So if this is a group of 100 people, then we would have to go through 100 people to find just 3 who is going to have biomarker positive. So to be able to do the stats properly, that tells us that we would need to look at 3,000 people over 60 to find the number of people who have the biomarker positive and then follow them to see whether they get Parkinson's disease or not. So what we are doing at present is we're beginning, so that's what the stats would have to look like. So we're trying, the reason we're doing that is that if we found these numbers, that's 3,000, there's a speller there, but if we found people in this stage, why would that matter? Because at this stage we haven't got a drug for treating it or slowing it down, but we do it because trials are more likely to work and we're more likely to find whether the drug is actually effective in slowing down the disease. Why is that? Well, it's much easier to slow a disease down when there's only 20 or 30% of cells going than when you're down here with 70% gone. The closer you are to the start of the disease too, you're actually able to say, well, what's the difference between people at this stage, 10 years before the disease? Can we understand what are the risk factors that they have and what's different about them to the people who don't have the biomarker present? So it's much easier to understand and study and identify what are the risk factors that might be at work at producing that factors. Is it true, for example, that people who are at the very early stages with a high biomarker, if we got them to really exercise, lose weight, starve a little, would we make the biomarker go back to normal? We know that that's true for other diseases if you get the biomarker early on. Would it be true of Parkinson's? Well, we don't know until we get there. So we're at the stage now where we're doing a pilot study, which is we're looking at 300 people aged over 60 to see whether in fact our biomarkers are high in the right proportion to actually justify going and looking at 3,000 people. And we need to do this sort of thing because quite reasonably granting bodies aren't going to give you the money to go off and look at 3,000 people if your biomarker doesn't hold up. They want to see the preliminary data before they spend a lot of money to do that. So Holly is going to be over here, and if you are this unashamed advertisement, is if you are over 60 and have no signs of Alzheimer's disease, Parkinson's or other, to your knowledge, you have none, and you don't have a problem in your shoulder, you haven't played football in the past and damaged it, and you're willing to give a pinprick of blood and wear a device on each wrist for 6 days, then we would love to speak to you. And I think Holly here will take your details and talk to you, but I think we'll do more on Monday next week, but also you can talk to her and negotiate a time if you're interested. So I'm willing to take some time from talking about questions, and we'll perhaps finish in about 10 minutes.